Okay, so what we're going to cover today is I'm going to introduce you to Poster Builder, which is that new feature I just mentioned earlier. A quick recap of the basics of BioRender that I'm going to do fairly quickly, just a little faster pace because most of you are hopefully already using BioRender. But if not, we'll do a quick recap. If you have any questions while I'm talking, please let me know. You can type in the chat or you can just unmute and interrupt me. Don't feel bad about interrupting me at all. I'm happy to answer questions live while we're in person like this, well, virtually, but answering the questions live will be quite helpful. And then I'll be sharing some advanced tips and tools that will hopefully save you a lot of time. So some highlights will be our multi-select tool, our alignment tool, customizing shapes and lines, and our connector lines and custom colors. So let's get started. And what I want to highlight to start is we've actually created pre-made Queen's academic posters in our poster builder. So you'll notice your logo is already preset. This is the preset blank version. Hopefully you would fill it in, and I'll show you how to edit poster builder in just a bit. But this is the classic landscape. And if you have feedback or you want to change the design in any way, we can change this quote unquote master template, but it also gives you a good starting point with the colors similar to your logo colors to kind of help with your research. So this is the landscape, and then this is our current portrait version as well. So you can actually access this yourself as long as you have BioRender Premium. How you do that is I'm just going to exit out of my screen. So this was the presentation mode in BioRender, which is using an illustration. So there's two things you can make from your main gallery. So this is the main gallery when I log into BioRender. You can choose to either create an illustration or a poster builder. For a lot of the presentation today, I'll be in an illustration. And what an illustration is basically a blank canvas, but you can also add slides. So you can have multiple and have this sort of like slideshow as well. And so if I go to poster builder right here, this is where I can actually find our Queen's template. So there's a couple other schools, as you've noticed as well. But if we just scroll down to sort of our popular templates, we did a lot of the main Ontario schools. And so if you just scroll down, you'll see a couple others as well, but you'll eventually see the Queen's University template right here. So it's right after the McMaster one. And so you'll see this is the landscape that I showed you, and this is the portrait that I showed you. And so I'm just going to click the Queen's one right here for you to see. You would select this template and then click Use Template. And what it's going to do, it's going to load this template in Poster Builder. This works a little differently from a classic blank illustration in that there's these nodes. Unfortunately, no matter how much I zoom in, the nodes don't get a lot bigger. But if you can see, I'll just hover my cursor near it right now. There's these oval-like shapes right here. And these oval-like shapes, I think of them as nodes, and they're edit points for you to either decide how large or small you want a section to be. So you'll notice I'm moving this to change how large or small my overall heading is. The poster builder will give you some warnings if it doesn't look great or if something's not going through the full header. And you might look at this and say, oh, but I actually want to add more figures in my results. I don't want to just have these slots right here. And you can actually insert content at the top right here. And then if I insert content, I can choose either section, title, and those are these big sections with a header, or just a specific content piece here. So potentially I just want to add an image in results. And as you notice, as I hover this image tile that I've dragged in, you'll notice it's adding a row at the top, or I can add it to the left. I can do just the left top row. So you can really choose where you want your image to go as well. I want to have an image in conclusions. So I'll put that in there. And then you'll notice it's automatically generated this box, this little section box. You can then move it using the nodes. It's also generated a caption box to remind you to make sure you have your caption. You could delete it if you don't have a caption as well. So it's very easy to just delete these slots. And then you're just using the nodes to adjust your shape. Poster Builder connects to your BioRender library. So if I click image right here, this was actually the library in the beginning. So if I had a signal pathway that I created in BioRender, I can click that and it'll actually load in the slot here. And BioRender will automatically scale it in a way that keeps the quality good and high. And if I move different nodes, it'll also scale the image accordingly. So just moving the nodes is enough to organize your shapes and different things like that. If you have figures that you might not have made in BioRender, you can actually upload your own files as well. So you'll notice I uploaded some Queen's logos, and you can actually upload anything that is a JPEG, a PNG, an SVG, or a TIFF file. So any of those files you can upload into your poster, and then you can have those figures under the image section here. If you work with a team collaboratively or your PI with other grad students on your team, I highly recommend making folders for your teams. And then you'll notice these are all the folders I have in BioRender. If I share these folders with everybody, anyone who makes an edit, adds an image, anything like that will have access to that folder. And so if I use this folder to work in my team, so I have a BioRender science communication team, I can actually see all the work that has been done in our communication team, and I can select one of these images from those teams. This is actually very useful as well if you have any grad students who might be graduating. And so for you to not lose access to their images, you can have them in the folder there. And so even if the owner of that canvas potentially goes to a back to a non-premium account, or they graduate, congratulations, you still have access to their work and to the figures they created during the time they were working in your lab, because they're in that folder. And so that's this poster builder here, where you can really play around to do a nice academic poster. I've also had librarians actually completely change these titles, make headings larger or smaller. And so you can actually adjust the height here, and using this to make infographics. So using this as a starting point to create different infographics or posters and advertisements, just because they found just clicking around with these squares a lot easier to navigate. And so there was a lot of utility in that. If you have any questions, feel free to pop them in the chat. Let me know. If not, I'm going to assume there are no questions on this. So this is our new poster builder in BioRender. Okay, perfect. Then if there are no questions for our poster, if there are, feel free to take your time to type and I can go back as I continue. But these are the main posters we have for Queens right now. And let's recap kind of the basics of BioRender as a whole. And I like to think of BioRender as us giving you a box of Lego. And so instead of a box of Lego, you're getting an entire library of different icons. And these icons, there's over 40,000 right now, but we update our library weekly. So we do get new icons all the time. And so think of all these icons as these building blocks that you can put together to make these cool images or different structures. Depending how you're combining them, it might be a graphical abstract, it might just be a protocol, but you're taking these pieces and putting them together to create whatever you need to create to communicate your work and research. And we have something in our library called templates right here. And in our library, we have something called icon packs. And what I really like about icon packs, so here's a bunch of icon packs. This is our template, and I'll highlight a couple here. What I really liked about them is it kind of shows me what we have in our library, especially by the topics. You can actually search things up as well, but I sometimes like to just see what is out there, and kind of see what I can use it for, how it's been used. And I also kind of like it, just sometimes I'll like a certain icon and I'll say, oh, I use benzene a lot. So I'm going to hit favorite and save it to my favorites here. And it's actually always going to be available. So now I have that benzene ring always available here under my favorites. And so I'm not having to go through the icon library every time I'm looking for a benzene ring. And so sometimes I like to just go through icon packs and see what I might use a lot, and then favorite different components so I can piece it together when I'm actually making a figure. So that's pretty cool. Those are our icon packs. But Going back to that basic building block in our library, what we have is we always start with our regular icons. And I can identify something's a regular icon with this nice blue box around the icon here. So notice the blue box here. And with these regular icons, you can change the color like so. So I have a bunch of preset colors here. Those were specifically done by our scientific designers to actually pick colors that are aesthetically pleasing to our eyes. So you'll see for different types of icons, we'll have a different number of presets as well. So this is our T cell, and you'll see there's a number of different presets for those colors. As well, you can actually add some glow to something as well. So if you want to add emphasis to a cell, or you're working in, say, a microtumor environment and you want to show a heat-like feature, adding glow here really helps layer your icons. And if you want to bunch a bunch of cancer cells together, let's say, adding that whole layer there using the transparency function can give you overall layers as you layer multiple icons on top of each other. So these are our basic regular icons. You'll often hear us referring to it as simple icons, because what we can do, like Lego Box, is piece simple icons together to make grouped icons. And so that's our next type of thing you'll see in our library, but you'll notice there, as I hover over this, you'll see a purple box. And that purple box identifies a group icon, which is different from this blue box of a regular icon. And with grouped icons, it's kind of like sticking two Lego pieces together. You've attached pieces together, and they're now this package that can be moved around together. They can also be resized together. So it's kind of this nice package. But if you wanted to edit individual components, it's still made out of the simple icons. So I can double click into a grouped icon and actually edit the individual components. So you'll notice I just edited the mouse. I could say move the tumor around or change the tumor color as well. So you'll notice I can edit the individual components, but I can still also move it around or enlarge it as a group. If you look here, this also with a bunch of cancer cells together, this is what I mean by you can change the color. You can have different transparency. So you'll notice in the back, it's a little more transparent, and that's adding depth. I potentially might want to highlight one cell. And so you'll notice I increase the glow on just the one cell there. And that's really something you can do for the grouped icons right here and still edit them as a whole. And no matter how big or small I'm making this, you'll notice the quality stays excellent. And that's because these are all vector-based files. So no matter how big or small you make your icons, they still look nice. And you'll notice this shape here is similar to this right here. And what this actually is it's still a grouped icon made out of simple icons. So I can individually change the color. But what's important to recognize is this right here is a simple icon. So just because they look similar doesn't mean they are the same thing. You really want to pay attention to where the boxes are here. And so it's that blue box versus that purple box if you can really edit those individual groups. So you'll be able to find grouped icons in our library. If I just searched up cancer cell right now, you'll see there's some icons that just exist with no sort of box. And if there's this nice purple box, that represents a grouped icon. So we have grouped icons in our library as it is, and you can just drag those grouped icons onto your screen. And the last thing we have in our library is our bio brushes. And bio brushes work conceptually just a little different in that they have nodes. So you'll notice these nodes right here. And these nodes, unfortunately, no matter how much I zoom in, kind of like Poster Builder, they're always hard to see. But I always think of the filled in blue circles as the exaggeration point. So you can play around here and really play with the overall curvature and shape with those blue circles. And think about the white circles as potential and emphasis points. So here I can make it longer, shorter, but what the white circles represent here is if I click this white circle, you'll notice it became a filled in blue circle. And now it's becoming a new exaggeration or emphasis point. So once you click that, it's going to generate two new white points as well. So you can really get the right shape that you're looking for by really moving around these blue nodes. And if you want another blue node for more customization, you just have to click a white node and it'll generate a blue node for you. And so our overall brushes aren't just limited to DNA. I wanted to show you typical, like we also have phospholipid bilayer. If you've ever drawn one on a chalkboard, they take forever to do. So I'm so thankful we have this in BioRender, and you can just change the color as well using our presets to get a color you're looking for. We also have a lot of vesicle or synapses, different things like that, that you can use within our brushes. So our brushes are right here in our icon library on the left-hand side, but you can also search them up. If I searched up DNA, you'll notice, again, there's nothing here. So these represent icons. So this is an icon version of DNA. But if I go back to that DNA search, you'll also notice that some of these have that little brush in the left-hand corner. So what that means is that represents the DNA brush, which is slightly different from the DNA icon. So you have the icon here, and this is a brush that looks quite similar to this icon, except when it has a brush, it has those nodes, so you're able to edit the overall shape. And if you're really happy with your brush, so this is a brush right here. If you're really, really happy with it, what you can do is actually convert your brush into a grouped icon. And what that can do is then you can color individual cells like this. You'll notice I could change a color, or I can delete just one cell of my brush. So you can really customize aspects of it. What you're doing when you're going from a bio brush to a grouped icon is you're changing from this node-like structure to overall making this out of individual Lego pieces, individual simple icons. And once you convert a bio brush into a grouped icon, you actually can't convert it back to the brush. So I always recommend, before you convert it to a grouped icon, hit favorite at the top. What it's going to do is it's going to favorite this in our favorite icon section here as that curved brush. So if I still need that curved brush, I still have access to that node version. And so if I made a specific shape, I'll still have that. And so here I'll click this. To make it a grouped icon, all I have to do is press the button, separate brush into editable icons. So I'll click that right here. And then you'll notice it's warning you that once you put it into a grouped icon, you can't go backwards. So it's warning you that you're going from the nodes to being able to have that purple box and edit the individual pieces. So I'm going to hit confirm, and then you'll notice I can edit those individual pieces like so, and then like this. And so you have individual pieces edited. But say you're like, oh, I actually don't like this shape. I want to change up the shape that I did. You can actually go back to favorites if you did remember to save it as a favorite, and you'll notice that I still have that brush here, and I can change the overall shape. So I've changed the shape as well, just because I saved it to favorites. So I always recommend hit favorite if you're planning to change it to a grouped icon, just because you can't go backwards from there. And so if you ever want to edit the shape in the future, you give yourself that option. And the last thing in our library are templates, and templates are pre-made by our scientific designers. What I really like about templates is I'm not a great artist to begin with, so it's always nice to see how other folks are designing their posters, or how they're working on different aspects of their figure, different ways they do titles even. And so I'll just show some general figures here of how they do the step-by-step processes, or the way they do different gradients as well. I often like to take design tips, but you can also use templates yourself. So you can actually use a template to add to it, or as a starting point, anything you'd like. If you create a lot of images and you actually want to get some scientific designer tips, you can actually submit your template suggestion as well. And you can either be a scientific contributor to it, or you can be the person who actually creates the template and get some feedback. We do also have some templates that are kind of empty, and as a starting point, my favorite example to show is we do a lot of timeline work. And so I just typed in timeline, and you'll see all these different types of timelines in our template library. And so depending what you want your timeline to be, or what look you're looking for, you'll notice there's a lot of different types of timelines here that you could use for your work. So that could be helpful as well. So feel free to use our templates. But those are the basics of what's in BioRender. So let's dive into our advanced features. And I'm going to start with our multi-select tool and our alignment tool. So in this slide here, you'll notice there's a lot of antibodies right here. I'm going to click this right here. When you click an icon, and there's multiple of that same icon on the canvas, the select same icon button will actually appear at the top of your screen. So I'm going to click this, and what this lets me do is it selects all the same icons in one go. And if I need to change the colors, anything like that, I'll be able to change the colors super quick with just one click. And then also if I wanted to say align them, I can say select same icons. So I've selected all of them, and we have an align tool at the top. I personally found it a little confusing in the beginning, and I always say the best way to look at the align tool is to look at that solid line. So you'll see a solid line either at the vertical left here, or vertical middle, or vertical right. So it depends what you're looking or how you want to align. This is down the middle horizontally. So follow that solid line to know where it's going to align to. So in this case, not a design tip, but potentially I wanted to align all the antibodies to the middle of the page down the horizontal. I'll just click that right here, and you'll notice all the icons are now moved to the middle of the page down the middle. But I'll show you some examples of where align and select same icon makes sense other than just this example. So let's look at a microtumor environment, and I'm going to bring in some themes from storytelling as well, just to really level up your figure design. And so this is a tumor microenvironment, but we also like to bring in overall storytelling themes. And so this is image from Star Wars, but overall in a lot of different stories and fiction, movies, things like that, you'll notice that villains are often, not always, but they're depicted as this reddish tone or an orange tone, where protagonists will have a green or blue tone or a more cooler color tone. And so we can apply this to scientific images as well, where if we kind of put cancer cells or our quote unquote bad guys in the body in this reddish orange or brown color and have the good guys in a cool green blue color, it kind of gives an overall story to what we're talking about when we're creating figures. So let's look at how that works in here. So when you have a microtumor environment like this, sometimes I just honestly bring the icon onto the side of the screen like I've done here, and then click select same icon. So it's quick, it's easy, I have access to it. And then I can just really quickly change all the colors in one go. So I'm gonna make all the cancer cells a dark red. And let me, for the dendritic cells, I'll click select same icon, and let me make them all green. And for the macrophage, select same icon. I'm going to make them all this blue color. And then it gives us this look. So you'll notice this overall kind of look of like, okay, this is a hot area. This is a tumor microenvironment. And we can see there's also contrast on what cells are working on this tumor environment. This also works with our cytokines. I'm going to call them cytokines, but in our library, it's actually called sphere. But you can select same icon, and you notice it select all the spheres. So if these were, say, a positive thing, I could change it to a nice blue color as well, as they're sending signals into that microtumor environment. And you'll notice that even though I selected that blue color, it kept the shading. So if you had any transparency changes that you were doing, it still keeps that color palette, but adds that bluish tone that you were looking for. And now it really gives that image the look of what is happening in this moment in your figure. And something else we also think about in coloring, I know this is a bit of a back to basics elementary school art class, but we also want to think about complementary colors or opposite colors when you have something of interest or you have something you're trying to contrast in your figure. And so looking at this color wheel, really follow those lines across. So if you have a red color here, you're really going to have a lot of contrast with green. Or if you have a blue color here, you'll have a lot of contrast to orange. Purple will contrast to yellow. And so we like to put these opposite colors next to each other to highlight and add a bit of boldness to your figures. And so here's a couple logos where you will actually see the complementary colors at work. So you'll notice that kind of blue and orange, or a yellow purple tone, or a green and red tone. And so we can apply that to our scientific images here. And so right now, the entire canvas here, it's got a kind of bluish color, bluish gray color to it. And we have a protein of interest. So if I did this entire figure, I can select same icon, select all the proteins on this page, and then change it in one go to a nice orange light color. I'm probably going to change it to this. And you can change it to a nice orange color, and it adds a bit of pop to your image. So you now notice that the protein stands out, and it really does look like it's the protein of interest because it's popping out from the screen, and that it gives a little more boldness to the overall look. And so I'm also going to show you the align tool as well. So this is different ways I've used the multi-select, but here I'm still going to use the select same icon because I want to get all the red blood cells nicely in the middle. So here's my align tool. If I want it in the middle, I'm just going to follow the solid line. So this is a solid horizontal, and click that right here, and you'll notice everything's nicely in the middle here. And then you can actually hit our distribute buttons as well. And think about distribute as how you want it to look, how you want things to be distributed. At this point, I want even spacing between each icon. So I'm going to hit this distribute button, and you'll notice it's nicely distributed the red blood cells, which is awesome. This is also helpful if you're doing like a lab protocol like this. And so I'm just going to lock the background. And what I can do is I can actually select all the shapes here, and I can then align it down the center like this. When I always look at this, I'm like, oh, this looks good. Everything is kind of in order, but I've noticed that I don't have a good eye for distribute and to make sure things have even spaces. And so I'll actually hit the distribute button, and you notice when I hit it, the boxes move just ever so slightly. And what's awesome about that is just moving it ever so slightly just helps keep things nicely in order, but also nicely distributed between each other. It just gives an overall cleaner look. And so that's the multi-select tool and the align tool. And I'm going to move on to our customization tool. So sometimes you'll be in BioRender and you're like, oh, I'm really looking for this shape, or I want to make this kind of shape as a background, but I can't find it in our library. So it's titled Custom Blob Tool, and I'm laughing a bit because that's a bit of an internal name where the official name is Custom Shapes right here. So if I go Insert Shapes Custom, that's the official name. But because it ends up as kind of a blob, we call it Custom Blob Tool. It's kind of a for fun name we say to each other. And so with the custom blob tool, I'll just bring it into our screen like this. It works a little like the brushes where the blue are these emphasis points, and you can really create exactly the shape you're looking for. So you'll notice I can really edit what I want the shape to look like. I can change the border width, so I can make it super thick. Or I can get rid of the border width as well. If I just want it to be an overall background color or backdrop, something like that, I have this available as that custom shape. And so with the custom shape, if you don't like any of these starting points of a custom shape, you can also freehand your custom shape. And so I'm going to freehand right here, and the lines look a little rough, but once I let go of my mouse, you'll see it's nicely kind of cleaned up this overall look. So you'll notice the shape has kind of cleaned up and it's a nice smooth line with all these nodes that I can edit. Right now, it's an individual line that I could potentially change the color of, but if I wanted to connect it to create an actual shape, an enclosed structure, I can actually, as I hover closer to this end here, you'll notice it'll hop on and connect. So I can actually connect this, and when it connects, the computer detects, oh, you've created this filled enclosed shape, and it'll give you color options to actually fill it in like so. And so it'll let you kind of play with the border and play with your overall stroke as well. And so when you are playing with this, it's a similar concept to the brush, as every kind of blue dot is this emphasis point. But if you want to add a new emphasis point, you can use any of these white dots here. And what will happen is if I click this white dot, it becomes a blue dot, and it creates two new white dots in between as a midpoint your other blue dots. So you can actually add these emphasis points as well. So this is the custom shapes, and some actual uses that we tend to use the custom blob tool for is one, emphasizing parts of a brain. So you can use this in different organs to overall show what you're trying to map out. And let me just add this here. A pro tip I got from somebody else is when you're trying to map out a specific structure, as I'm trying to show with the brain, something you can do instead of trying to match it to the lines is increase your transparency here. So when I increase the transparency, I can very much see, oh, I didn't capture this curve right here. And so if you increase your transparency, you'll know a little better how well you're encompassing parts of that organ. And so I'm going to do that right here, like so. And so say I wanted to just capture this part of the brain right here. I've got that shape, that structure's filling it in right now. Perfect. I can bring the transparency back down so it's an opaque look, and then I can change the color to whatever I'd like. So potentially I'm going to change it to this navy, and then every section of the brain, I'm going to use the blob tool to model off a different color. And so you can really choose to overall emphasize aspects of an organ, things like that, using the blob tool. Or if you're trying to show a protein and you can't find the right structure, you can actually use blob tool to create a protein of your choice and add some genetic material. And then here we now even have our CRISPR protein. So that's pretty cool. I can even purposely group this. So I've selected both options here and I can hit group, and then it'll group this all together. So now I have this as one piece. And if this is something I'm going to use a lot, again, hit favorite just so it saves to your favorite on the side here, so you always have access to it. Some cool examples of where blob tool has also been used is right here with our scientific designers. So the cross section of the spine and the brain. And what I'm just going to do here is I'm going to bring this all in to overall show this structure right here. So you'll notice I'm bringing in the blob tool here, and then the blob tool has just been used like all around here, all these shapes that are highlighting aspects of the spine here. This is all just the blob tool being organized well enough and done nicely to emphasize that part of the spinal cord or the brain. You can't delete nodes, unfortunately. So once you have that blue node, it does permanently exist. You can try to map. Sometimes you can try to, if you didn't like it, I sometimes try to map a node on top of each other to give an overall, and that will give like a smooth line look. So you notice I kind of mapped two nodes on top of each other here. They're very, very close. And that kind of gives a nice smooth look though, overall from afar. And if you're worried you've made too many nodes, hit favorite so you have it right before you make too many changes. And so here, another node, you'll notice there's a lot of nodes in here, and I'm making changes like that. So I've highlighted aspects of the brain as well, just using the different blob tool. And some have asked me, how does this line show up here? The line is not actually what is created by the blob tool. It's just that there's a bit of transparency. So some of the brain is actually coming through. If I bring the transparency all the way down, you'll see the blob tool as a completely opaque structure here. So you wouldn't see that line. So it's actually just a bit of transparency here where it's letting that line come through like that. And now you're seeing the line. So that's actually the background from the brain. Okay, so that's our blob tool, but some other custom tools we have that I find super duper useful is lines and arrows. I know that sounds kind of funny, but lines and arrows are super powerful. Our circular arrows are really, really powerful. The generic thing that you often see is people like, oh, I can do a plasmid map. True, you can, and it's quite useful in that your circular arrow, the nodes now represent start and end points. So you'll notice I'm moving it around, showing you it as a start or end point. I can increase my width. And as I increase the width, it becomes more obvious that there's this white node here. And white nodes in circular arrows are cut points. So I can actually cut this up right here, and it creates a new start or end points. Endpoint. You can also customize what's on the end. So if I don't want an arrowhead, I can change it to an inhibitor. I can change it to also a neuroreceptor, right? So there's different things you can actually change your circular arrows to, and that adds a lot of customization to what you can use it for. You can also increase the border quite a bit. So I'm going to increase it all the way. And I'm going to change this to no end here. And the reason I'm doing this is sometimes when you're doing a circular workflow or a circular pathway and you want to show step by steps, you can really use this because you can cut this and then say, okay, so this is step 1 of the cycle, and then move this on over and say, okay, this is step 2 of our cycle. And you can really use these circular arrows to create that circular cyclic pathway. And I'll show you some examples of how that looks. So quick mapping of a plasmid, we're going to just insert a circular line here. So let me just bring that onto our canvas. And you'll see I'm just trying to match it. We already had a pre-made gray circle. So someone had once asked me that. This is a pre-made gray circle just from inserting a circular shape. And I'm going to increase the width a bit as well, just so it stands out a bit. And you'll notice I have gene A, B, and C. So let's cut this up 3 times. So I'll cut it once here, bring this over as gene A, and then I'll cut this once again to bring this over to gene B. So all I'm doing is changing that start and end point along that circle. So it keeps that circular structure as well, but I'm able to cut it to be the exact length of whatever my gene might be. And what you can do with the arrows is actually customize each individual portion. So I've clicked this first one here and say I want it to match the color of gene A. I'm just going to hit color here and I can click color picker at the top. Color picker will give you a square here. And as you hover on different aspects of your canvas, it's actually going to detect what color it is. So I can actually match that first arrow to my gene A font. So that way I have this nice teal color that matches, and I can do the same with the rest. So I'm going to do that with the yellow-like color from gene B and get that golden yellow. And then I'll do the same for gene C so that I'm able to really map it out with the right colors as well and give that overall look. So you'll notice there, that's like a plasmid map that I've made, super easy, super quick, and I've matched the colors as well. And going to my next slide, these are just examples of ways we've used circular arrows. So this is what I was talking about with your phase 1, phase 2. All this is a circular arrow with a very, 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 very enlarged width. So you'll notice the width is quite large. And then people change the colors of that aspect. So it gives a nice gradient look. But this is really, at the end of the day, I can move this all around. It's just a circular arrow. So this is a circular arrow with no ends as well. Or you can keep the ends and do a gradient look like this. So they've inserted, this is called a insert text shape, where you can have different shapes with the text in there as well. And so that's what they have right here. But if I play with my arrow as well, so I have my arrows here, this is actually, again, that circular arrow, but it was just cut up a bunch of times, and then the color was changed to give an overall gradient. So you have a nice little gradient, but you'll notice that it changes. It's still all in that same pathway, but you've put text boxes in between and have that overall gradient look based on the colors you've used. And then lastly, here's a figure where you see the circular arrows in action. So again, you'll see different ways that circular arrow has been used. So I can click it right here and I have a circular arrow here. So there's quite a different ways you see people using it overall. And another time-saving tip outside of the circles is using actual connector lines. So connector lines are fun in that they don't have a specific button you click to make it happen, like the select same icon. But if I have a line here and I want to show a process where protein A is phosphorylated into protein A with a phosphate group, I might want to show this connection. As I select insert line arrow, you'll notice as I come onto the screen that it's identifying, the shape is recognizing, oh, you have a line right now. You can actually connect the line to the shape. And you'll notice that there's these white little dots that show up, and I can actually connect these lines to those little circles. You know it's detecting a connection when that small black line with 2 black dots shows up. That's the computer saying, hey, we're going to connect to this dot right here. And so I can actually connect to the dot. And as I approach this object here, you'll notice that the circles are appearing here, and I can actually then connect it. What it means when it's connected is I can move this shape wherever I want to move it. And you'll notice the arrow is following as well. So I can move things around my screen really easily, and then it's just following around as well. When I put this originally on there, I had clicked where there was a circle. So I'm going to show you again on this side where there's a circle here and I'm filling it in. But you don't necessarily have to always put it where there's a circle. With connector lines, you can actually connect it to the corners here as well. So notice the corners don't have a white circle, but what is happening is, it's hard to see, but you'll notice a small white line with the 2 black dots is appearing. So it's identifying, oh, you want to connect it to the corner. And so it's going to allow me to connect it to the corner. And now I have it connected to the corner here. And what I can also do is create nice little bends like this. And a small design tip we always recommend is instead of having angles like this, so here this is a slant, this is a slight angle, we always recommend to try to keep things to a 90 degrees angle. It just has a cleaner look, either on a horizontal plane or a vertical plane. Keeping everything either nicely horizontal or nicely vertical helps it helps generate an overall clean look for your figures. Not only do lines connect to text boxes, though, they also connect to icons. So here I've already pre-connected them, and you'll notice if I ever wanted to move this protocol, say up the screen, it's really just a quick click. And because they're already connected, it's pretty easy. Our alignment tools will tell us what to do. So I've moved that all up, and then I can highlight all of this to move it up as well. And so it's really easy to adjust aspects of your image if you have it all nicely connected together. So you can use those connector lines to connect things together. We also have a flow chart here where if you did connect things together, but you accidentally didn't align things nicely, if they were all connected, just moving a text box helps align the whole structure. And then going back to the idea of cleaning things up by having those 90 degrees vertical angles, what I'm going to do is I'm just going to line things up a little better. So instead of having this slant, bring it down so it's on a horizontal plane. And then similarly here, instead of having a slant, what I'm going to do is I'm actually going to move this node and bring it to the corner and add a nice 90-degree line. And I'm going to bring this to the middle here. So what I'm trying to do is make just a cleaner look. So it just looks less cluttered. And you'll notice now with the straight lines, the horizontal and the vertical, it just is a lot cleaner, easier to follow, and more organized. It just looks a lot better if you ask me. Okay, so another way we can customize is customizing our colors. So I already kind of showed you with our plasmid maps, but what I'm going to show you here is not only can you just color picker and pick what's on your canvas, you can also enter hex codes as well. So if I click this cancer cell and none of the presets are to my interest, what I can actually do is click the color overlay here, and I can enter the hex code that I want. So 173 F, oops, my keyboard does not like me right now. 173, capital F, 5F. I don't know why it's entering like that. Let me reset this. And then I'm going to try to enter the hex code again. 1735. Perfect. And so what it's doing is it's going to preset the different shades of the cancer cell to be in that color palette. So you'll notice there, I've entered this hex code right here, and I can click apply, and then I have that specific color right here. If I'm not a fan of entering the hex code, I'm personally not, because I make a lot of typos, I can hit color picker and just hover over the right color, and then it'll match the cell to that overall color as well. So either or, whatever suits your fancy, but I can also then add the hex code here again. So ED553B, and then it's going to match it to that color. So if you have specific colors you have to follow for a logo or for an ad, anything like that, you can use those colors. You can use a hex code. Sometimes people need a contrast between certain hex codes, so you can actually then use those hex codes as well. And then something to think about is we also have access to the PDB upload. So if you ever want a specific protein structure, we can actually make an icon using the protein database. So here is the green fluorescent protein on the COVID virus. And so even if I wanted to match it, I can go select same icon. It selects them all. I can change the color real quick and then add some glow as well, and to add that kind of glowing effect. And then it's layered. So there's two icons here. You'll notice we have the secondary structure ribbon pattern here. And this here is just the 3D van der Waal 3D structure. But you'll notice as an overall design tip, which I really like, is they took the 3D structure, but they increased the transparency, and then they layered a opaque version of that secondary structure on top. And so you can actually grab both of these from the PDB. So if I go to PDB retrieve, I just know the SARS receptor protein off the top of my head. So I'll type that in. The code is 6VXX. You type it in and you can actually load the protein in the PDB, depending which view you want. And you can pick, so this is the quick surface, which will show the overall 3D structure. And depending what you want, you can angle it to save as an icon at whatever angle you would like. You can also change the colors as well. So you can change how you want it to look. You can add a border to it as well to change that look as well, and play with the overall idea of what you want your protein to look like. Once you're happy, say I want a bird's eye view actually. So I want what it looks like from the top. This is what I want my icon to look. Depending how you want it, you can click save as new icon. And then it's going to save right up top here and you can drag it onto your canvas. So it's actually a cool way to bring in the PDB or specific protein structures that you're looking for. And last tip is collaboration. So you've probably noticed there's this nice share button at the top, and I mentioned it with folders earlier in this webinar as well. I recommend always sharing things between your team because that way everyone has access to the work that you're working on. You can give people either view access or edit access. You can do it through either copying a link or actually adding their emails. If you have a team set up, you can also invite all the members of the team that you're on. But here I can give you all view access to the slides. So I said anyone with the link can view. I'm going to copy the link and put that in the chat. So that way you all have access to this slide deck. You can't edit it, but you can view it at any time. So that's a really good way to collaborate with your peers or also to do a presentation and share it. You can share posters as well from our poster builder. So super easy to share your work. And last but not least, I always say to have fun in BioRender. We also have our Biomojis in our icon library. And so you'll notice I've made this Biomoji and actually put it up on my screen here. But you can actually go to our library and click Biomoji, different items here. So you have different faces, you have different cells, you have different items that you can add. So potentially, oh, I love bananas. So I'll add a banana here to this little biomoji right here. And what I can do is add different options. And if I'm happy with how my biomoji looks, notice I've selected it all. The button at the top here comes up as save as my avatar. So now I can actually save it. And it's going to, in a bit, appear right here. So now you see my little green cell holding the banana. And so you can actually create your own biomoji in BioRender as well, which is always fun to see. So always enjoy what you're doing as well in BioRender. It's a lot of fun and there's a lot of cool things you can create. Once you get really good at BioRender, I always like to show this one in the end. This poster right here contains a lot of elements that we talked about today. You'll see the PDB with some transparency factors here. You'll see a custom shape right here with all the nodes, these nice clean 90-degree lines, as well as down here. These are some custom lines. It's just the custom arrow stuff I had showed you before, but you can specifically choose to have nothing on the end and a circle. on the end. And so those are ways you can customize the lines to be what you would like. And really how you put things together can really create a really strong aesthetic image. And so here again, that custom shape, you'll notice all the nodes here, all the nodes here. You can find this template in our template library. So if you want to dig deep a little more, I liked how they just even used a line as a title here. So if you want to see more of how they just use these design tips, you can just look up 2020 Nobel Prize in our template library and you'll find this right here. But I like to click around and even see like, okay, so when did they increase transparency? How did they create depth? How did they do layering in here as well? And so overall, looking at how this was done, I find pretty helpful. All right, so I'm sorry we started a little late, but it's now 11 o'clock as well. So I want to make sure we're not going to overtime. So I just want to thank you all for joining me this morning and learning a bit more about BioRender. Feel free to reach out anytime if you have questions, and Brooke will be your go-to for anything in terms of the licensing. And we can always stay in touch.